Welcome everyone to today's webinar on using cover crops as a carbon source for anaerobic soil disinfestation, or ASD. I'm Alice Formiga of eOrganic. This presentation will last about an hour, followed by 30 minutes for questions. So today we're going to learn from two of the team members of a NIFA OEI-funded research project who are learning about how to optimize anaerobic soil disinfestation for organic production in different regions of the United States, Francesco Gioia and Kathleen Arrington of Pennsylvania State University. This is the third webinar in a series from this project, and you can find both of those webinars on the eOrganic YouTube channel, as well as by Googling webinars by eOrganic and finding them on the eOrganic website.
Good afternoon or good morning, depending on where you are. Today, this webinar is focused on the use of cover crops as a carbon source. As Alice mentioned before, we have done, some webinars before. This is part of a webinar series. And beside the first webinar, there was an introduction to ASD. Last month, we did one that was focused on the use of, in general, selecting carbon sources and defining the application rate. And now cover crops can be used as a carbon source for ASD, but we thought that it would be good to have a dedicated webinar to discuss this topic. And so that's what you have here today. And we are going to probably have more webinars in the fall. We will have kind of a break, considering that a lot of people are busy with the growing season and will be busy as well. But in the fall, we are already preparing, for the fall to come back with more webinars. I just wanted to remind that this is for anyone that is new to this webinar series, that this is one of the products of a project funded by the USDA NIFA OEI program, and that is focused on ASD and involves partners from Penn State and Penn State Extension, as well as USDA-RS, in particular the U.S. Horticultural Research Laboratory in Fort Pierce, Florida, and then colleagues also from UF-IFAS in Florida and Oregon State University. And we have a web page on the eOrganic platform that you could visit if you're interested to find more information about the work that we are doing. The project per se is focused on optimizing ASD for application in organic vegetable production systems, but we are also including strawberries in our work. And we are conducting most of the work in two regions, like Northeast and Southeast. So we are working in Pennsylvania and Florida as representative state of these two regions. And as you can see from this layout, we have this is an interdisciplinary project, and we have a series of objectives that we are working on to kind of look at anaerobic soil disinfestation 360 degrees. So please, get information from our website and reach out to us if you have any question. Here, I'm gonna briefly show the outline of the presentation. The goal of this webinar today is to provide basic information on the opportunity to integrate the use of cover crops as a carbon source for the application of ASD in organic vegetable production systems or horticultural systems. And so we will have a brief introduction, very brief, on ASD and cover crops, and then we will dive into how to select cover crops that are suitable for ASD application as a carbon source, the factor that influence the efficacy of cover crops as a carbon source. And then my colleague, Kathleen Arrington, here from Penn State as well, she will present a case study on cover crops characterized by different carbon-nitrogen ratio that we have a 2-year study that is almost complete. And then we will do some concluding remarks, and then we will start with the Q&A session. So I'm gonna stop my video here so that you don't get distracted by my face, and we will start with the introduction here. So let's see if I can move this. I'm having some trouble here controlling the slides. So for anyone that didn't attend this webinar series before, I just wanted to provide a brief definition of what is anaerobic soil disinfestation. We define ASD as a biological solution for the pre-planting management of soil-borne pests and pathogens. And that includes fungal and bacterial pathogens, plant parasitic nematodes, and weeds. And usually is applied to horticultural crops because for other crops, it may not be a cost-effective solution in managing soil-borne pests and pathogens. In the last webinar, we also went through how ASD is applied, and it is a three-step process. The first step is to amend the soil, incorporate in the soil readily labile, decomposable organic amendment. Ideally, the carbon-nitrogen ratio should be 20:1, and this essentially is providing carbon sugar to activate the microbes in the soil and start the process. Then what we do is to uh, use like a tarp or a film that usually is a total impermeable film, and this helps to develop anaerobic conditions and maintain those anaerobic conditions over time. And finally, the last step is to irrigate the soil to saturation or field capacity if you want. And this is like really meant to help the development of anaerobic condition and then promote the diffusion of byproducts that are generated during the anaerobic soil disinfestation treatment. So in the last webinar, we focused a lot on carbon sources. We went through what are carbon sources suitable, what are the characteristics of those carbon sources, and what are the criteria we use to select alternative carbon sources. And of course, availability and cost are some of the main one, but then also the physical, chemical, and microbial properties are important. And we discussed all of this in the previous webinar. And in the previous webinar, we also mentioned that cover crops can be a suitable carbon source. And so today we're going to focus, we didn't say much last time about cover crops, and we're going to focus on cover crops today. What you see here is a nice picture of some snow peas that are in a mix with some grasses and radishes. And it's like a very common cover crop mix that we use as a like overwintering cover crop here in the Mid-Atlantic region. Now, what are cover crops? Why do we grow cover crops? Since like many of you are familiar with cover crops, I limited this to just one slide. Cover crops are plants grown in rotation with cash crop, and the primary purpose of using cover crops is to keep the soil covered. And the reason we do that is because keeping the soil covered, we are like with cover crops, we are trying to improve soil health. Now, beside improving soil health, cover crops may have other benefits. And, for these benefits are, for the entire agro ecosystem, but also for the following crop, the crop following the cover crops. Now, when we talk about these multiple benefits, we can see here in this little scheme here that cover crops are used primarily to increase organic matter and sequester carbon. They can increase water infiltration, promote water retention, reduce soil compaction, sustain, and provide food for soil life, all the life that is in our soil, reduce nutrient loss, reduce soil erosion, suppress weed, provide nitrogen, especially for leguminous that have the ability to fix nitrogen, atmospheric nitrogen, retrieve nutrients from lower level of the soil. And some cover crops have also provide as a benefit the opportunity to manage plant pests and pathogens. And then some also can attract, sustain beneficial insect. Now, when we are selecting cover crops for our normal organic or conventional production system, what we are doing is like identifying what is our priority, what is that we want from that cover crop, and based on the goal that we have, we will select a cover crop or a cover crop mix. And in this case, we are talking about using cover crops for as a carbon source for ASD. And what that implies is that we are still considering all these multiple benefits that can provide, that cover crops can provide, but we are looking also for some characteristics of the cover crops that will maximize the efficiency, the efficacy of the ASD treatment. And so we will discuss now what are those characteristics and how we select cover crops. Now, before we do that, the main question that we had when we started working on this was, is it possible to integrate the use of cover crop and anaerobic soil disinfestation? And the answer is yes, we can do it. And we see like cover crops and anaerobic soil disinfestation as two practices, two biological practices that have as a common objective enhancing soil health. And using cover crops as a carbon source for the application of ASD may allow some synergy between these two practices. And an example that I can make there is if, for instance, we are trying to control weeds with anaerobic soil disinfestation because we had a weed issue, we could select a cover crop that, while the cover crop is growing, is already having an effect in reducing the infestation of a specific weed or a group of weeds. But then ASD is like cover crops is also used as a carbon source, and ASD will have, in addition to the effect of the cover crop, another effect on suppressing weeds. So the way we see this is like really integrating two approaches to control better weeds, for example, but this can apply also to soil-borne pests and pathogens like nematodes, for instance. There are some cover crops that will control better nematodes than others. So this is what I wanted to mention. And what are some of the advantages of using cover crops as a carbon source for ASD application? The first advantage is the opportunity to produce the carbon source on site on our farm at a relatively low cost. And this is important, especially if we are trying to have kind of almost like a closed cycle, where we are not taking much input from outside, and we want to generate the carbon source on our farm. Then, the other advantage is the opportunity to reduce the need for other organic amendments. So sometimes the cover crop may not be enough as a carbon source, but definitely the fact that we are using a cover crop may reduce the amount of other organic amendments that we may need for the application of ASD. And then the other aspect is the opportunity to combine the effect of both practices. And this is true, like for the example I was just making of soil-borne pests and pathogens, but also weeds. And and then there is another aspect that is interesting is the opportunity sometimes to use the residual fertility that we have after the ASD treatment, after the cash crop. So we apply the ASD treatment, we can have a crop, and still there is residual fertility that derives from the ASD treatment. We could use that residual fertility to establish a good cover crop and start the cycle again. So this is like, some of the advantages that we have when we apply ASD with using cover crops. Now, the other question are like how to select cover crops suitable for ASD application and what factor determine the efficacy of the cover crop as carbon sources. What you see here in the picture is a cover crop of buckwheat, just buckwheat. Now, when we are trying to select cover crops for the ASD application, as it is true for, generally when we are working with cover crops, the selection of the cover crops is driven first by the specific crop rotation system. And in our case with ASD, by the timing of application of the ASD treatment. And so here I'm reporting just some example. If we take like a full year, and our crop is like a spring, summer cash crop, like let's say fresh market tomatoes, we want to apply the ASD 3-4 weeks before we plant that crop. And so that means that this is kind of defining the crop cycle of that summer crop, and the application of the ASD timing is defining what is left for growing a cover crop. So what we see is, for instance, after tomato, we have the opportunity to establish overwintering cover crops. They will grow until mid-April or sometimes the beginning of May, depending on the cycle that we have, and we can terminate that cover crop using it as a carbon source for the ASD treatment. And so this will be, in this case, we will be looking at overwintering cover crops, which are different from cover crops that we could grow over a spring or summer period. And I'm making here another example with strawberry. Strawberry usually in the mid-Atlantic region are planted sometimes like early mid-August. And so the ASD treatment is applied usually toward like end of July. And before that, we have some, in some cases, we have the opportunity to grow a spring, summer cover crop, and that could be used as a carbon source. And then, you start the cycle of the strawberry, and maybe the following year, the cycle will continue until, July or August. But, there is an opportunity there to use summer cover crops. And here is another example. Sometimes we have fall winter cash crops. And, I'm making the case here that we are growing, let's say, a crop in high tunnel. And we can do the application all the way, by the end, like, let's say, the end of September, mid-October. But, usually to establish a good fall winter cash crop, we need to plant it around September. And so the ASD treatment is applied 3-4 weeks before that. And still here we have the opportunity to grow a spring summer cover crop that could be used as a carbon source. So in this sense, like these are like, we need to consider, we need to take into account the specific crop rotation system we are working with and the timing of application of ASD to understand which type of cover crop we are growing. Then we can go deeper into, the selection of the cover crops, considering the main characteristics that we are looking for in a carbon source. And when we talk about cover crops as a carbon source, these are the main traits that we are looking for. One is the capacity to generate a lot of biomass, fresh biomass, in a relatively short time. And the case here of the buckwheat is because really is a fast-growing cover crop that within 45 days, something like that, can really produce a lot of biomass. Now, besides generating a lot of biomass, we are interested, like for other organic amendment, in how much labile is the carbon, like how easy is to decompose that carbon. And then that is determined a lot by the quality of the carbon, the quality of the sugar that we have, and also by the carbon-nitrogen ratio. So these are important parameters that we are considering. And then we take into account the capacity to suppress soil-borne pests and pathogens. Like the example that I made before is, if our target is to control weeds, we want a cover crop that is going to be able to suppress weed by itself, and then ASD can add on to that. Or if we are looking for nematodes, controlling root knot nematodes, we don't want to put a cover crop that may be susceptible to nematodes, because that will multiply the inoculum. Instead, what we want is a cover crop that is going to contribute to suppress root knot nematode in combination with the ASD treatment. Now, to go through these characteristics and show you some of the work that we have been doing, and what we mean with those characteristics, I'm going to present like the results of a preliminary study that we did under control environmental condition with 12 different species of cover crops, including some leguminous that you see here, brassicas, and then buckwheat that I mentioned several times, and some grasses. And on this column here, you can see the broadcast seed rate that we use. Now, when we are trying to maximize the biomass that we generate in a given time, in a relatively short time, sometimes the seed rate, the seed density that we can use for these cover crops may be higher than what you normally will use in a regular cover crop, just because you are trying to generate a lot of biomass in relatively short time. And so these are the application rate that we use for this study. And this was a pot study conducted in greenhouse. And this was this is a view of the crop, the treatment at 32 days. And then this is what it looked like at 46 days. And here, what we did was to determine the cover crop plant biomass. We determined the above ground biomass at 32 and 46 days to see what can we do, what we can generate in terms of fresh biomass within this time period. And of course, what you can see here is that at 46 days, the biomass was substantially higher compared to at 32 days. And there is pretty good variability between the different species that we tested, and some of them had more biomass than others. We wanted to consider not only the above ground biomass, but also the root biomass. And so here is some picture of, oats at 32 days and forage radish at 46 days. And the idea is that, this poster here is pretty famous, is that beside the contribution of the biomass that we can generate above ground, the root of the cover crops can have a big impact on our soil health and a lot of the function. But in this case, even in generating biomass for the ASD treatment, because we can incorporate even the roots into that as a carbon source. So in this table here, what you can see is kind of the summary of the total plant dry biomass, but we have the root and the above ground dry biomass. And the main thing that emerges here is that buckwheat had the highest total plant dry biomass, but there were also other crops that were really interesting and they were right behind it. Now, what we did was, besides assessing the biomass, we analyzed those tissues for the total carbon content, and multiplying the total carbon content by the biomass, the dry biomass that each cover crop generated, we estimated what is the total carbon applied for each of those cover crops. And we implemented the ASD treatment in actual soil using this cover crop residue. And what we saw is that when we looked at the cumulative, the level of anaerobic condition that we developed with each treatment, using molasses as a control, we see that there was a good relationship, a positive relationship, increasing, let's say, the amount of total carbon applied. It will increase the cumulative redox potential. Now, here we have all the cover crops, and these 3 points here were molasses. And so we continue to analyze the plant tissue samples, measuring also the fraction of carbon that was easily oxidizable using the permanganate method that normally is used for soil. We used it for cover crops. We adapted to cover crops. And the other thing that we did was to measure the total sugar content of the cover crop tissues with the Entron method. And what we observed was that when we use these two parameters, so when we use the total sugar and the easily oxidizable carbon fraction, the relationship with the cumulative redox potential, it was had a better fit. The issue here is that there was very big distance between molasses and all the cover crops. And when we look at the cover crops in comparison with glucose and cellulose, what we see is that they are all kind of flat here. Buckwheat stood out among all the other cover crops in terms of biomass produced, total carbon produced. But for instance, buckwheat had lower labile carbon than other carbon sources. And for sure, in this study, we didn't consider like the carbon nitrogen ratio. Now, last time in the last webinar, we talked about the fact that because we have a lot of cellulose in cover crops or plant material, the cellulose is not as degradable as, let's say, sugar, simple sugar like glucose or saccharose and things like that. And so that is kind of a limitation for cover crops in using cover crops as a carbon source. Now, the other thing that we have not considered in this study is the effect of the carbon nitrogen ratio. And we clearly saw that a lot of the leguminous had a better carbon nitrogen ratio, but they didn't produce enough biomass. Now, this was just a pot study. And so we were using that as a preliminary study to identify some carbon sources, that some cover crops that could be tested as a carbon source in the field. And what we did was during the same season to select 4 of those. We included buckwheat, forage radish, oat, and sun and we did a field experiment. And at the beginning, the field experiment, as you can tell from this picture, was looking really good. All cover crops were doing well. And in particular, you can see here some pictures with, so the detail here, this is buckwheat. Over here is sun and sun did okay, but it was not outcompeting weeds. So it was like an issue for sun weed were an issue. At the same time, oat was doing very well. It was suppressing all the weeds, but to a certain point, it was severely affected by rust, and so it declined pretty quickly. Forage radish, I think this was not the ideal season to do it because it was affected by drought. There was a week, 10 days of drought, and there was no irrigation for this. And then the plants were also affected by beetles that will chew on the leaves. And in the case of sun what beside the issue with the weed, there was an issue with Japanese beetles. So I'm showing all this to say that the idea of growing a cover crop as a carbon source is really good idea. At the same time, you may end up having certain issue, and the cover crop may not grow as you want, and that could be an issue. Now, among those 4 cover crops that we tested, buckwheat did really well and had no problem. It was really fast growing and very interesting. So what we decided to do, since we had only 1 cover crop left, was to test buckwheat ex situ, like taking the above ground biomass and using it as a carbon source by itself or in combination with molasses applied at half rate. And then we used as a comparison molasses full rate that you see here. And then we had an untreated control. We also amended the plots with the chicken manure. And we had 2 types of plastic, black total impermeable film and clear total impermeable film. So this is the way the buckwheat was harvested at this point, and we had extra strips of buckwheat that we seeded for to have enough material. And this is how after the forage harvested went through. This was the outcome, how it came out, the material. And these are the plots that we used to do the study. This is a movable light tunnel structure. You can see here the application of molasses diluted 1-to-1 with water, and then how we applied the cover crop residues on top. Now, this is like we don't get the full benefit of a cover crop, but we were just testing the impact of the material, the buckwheat as a carbon source. So everything was incorporated in the soil, and then we used the clear and black TIF film. We moved the eye tunnel on top, started irrigating everything, and started setting up sensors to measure the soil redox potential, the soil temperature, a bunch of stuff. We did a bunch of soil sampling and assessment on the plants. We were growing cabbage afterwards. And here what you can see is the soil redox potential, how anaerobic the soil went. So this is the time of application. The dashed line is like indicating anything below is anaerobic, anything above is considered aerobic. So presence of oxygen, absence of oxygen. And what you can see here is that the treatment that we tested really had an impact on the development of those anaerobic conditions. We can see here that ASD full rate applied like with clear film was at a longer period of anaerobicity. And what we do then is to calculate the cumulative redox potential. And this cumulative redox potential, what we can see here is that this is the molasses applied full rate without any carbon source with clear and black TIF. There was no difference between clear and black TIF in terms of like statistical significance. And what we can see is that whenever we use buckwheat by itself or combined with alpha rate of molasses, we got still very good anaerobic conditions, but it was not comparable to the molasses itself. Now, our threshold to say that a treatment is effective in terms of ASD treatment is this 50,000 millivolts per hour. So you can see that all the treatments except the untreated control were above that line. So the treatment was effective. And that, it was a demonstration that we could do ASD application with a cover crop and growing the cover crop as a summer crop and then doing the application in the fall. That application was done at the end of August, beginning of September. Now, there is still some question you may have, like when I apply ASD using a cover crop as a carbon source, is there any adjustment I need to do in the application of the technique? Also, are there challenges in using cover crops as a carbon source? What you see here is a sorghum Sudan grass cover crop that we used as a carbon source for another study. Some of the main challenges. So the answer to the first question is like, in terms of application, there's not much change, but there are certain things that we need to do when we are working with a cover crop. In terms of challenges, the main challenges that I see are, to identify a window of time that is sufficient to grow a good cover crop. One of the challenges is to balance the amount of biomass that is generated with the optimal carbon-nitrogen ratio and the soil conditions at the timing of the ASD application. And an example that I can make is sometimes the cover crop is wonderful. You have very good biomass and you are ready to apply the ASD, but it's raining for a week, 10 days, and you cannot do the application of the ASD treatment, and the crop continues to grow and continues to increase the carbon-nitrogen ratio that may change over time, and that becomes suboptimal for the application of ASD. And in that case, we need to adjust what we are using as an organic amendment, maybe adding a nitrogen source. The other thing that we have is that, so the challenge is variability of pedoclimatic condition. The outcome of growing a cover crop, it depends really by the environmental condition, the timing when you seed it, the timing when you're doing the application of the ASD treatment. And an example that I want to make here is like in this picture, you can see this is crimson clover, and then we have triticale and triticale as a mix, and the weeds are not controlled in this plot with the crimson clover, while they are very well suppressed when we use the mix or just the triticale. And here's like a detail of the plots side by side in comparison. Now, another challenge is the fact that often we don't rely only on one cover crop. It's a good practice, sometimes to use mixes. I would say often is a good practice to use mixes because, a cover crop could provide more benefit in one direction, another one could provide benefit in another direction, and the mix sometimes seems to generate more biomass, a better balanced carbon-nitrogen ratio, and so understanding, what is the right proportion of different species that you should mix together in your area, it's critical, for the success, and it's a challenge, but it's important for the success of your cover crop and its use as a carbon source. Now, in this set of slides here, I just want to show how we apply ASD using a cover crop like sorghum Sudan grass. Sorghum Sudan grass is great because it can generate a lot of biomass. Sometimes we can do 2-3 cuts out of the same cover crop if we have the opportunity. And in this case, you see the field, we use a flame mower, and sometimes we may go 2 times on the same section, the same area, to chop it up a little bit more. I think what you see in the picture here is the results of one pass. But if you do more passes with the flame mower, one against the other, you may chop it up more, and that, it could be beneficial. Then to terminate a cover crop like sorghum Sudan grass, you really need to plow the soil, otherwise it may start over again. And so this is what you see here in the picture. And in this sense, like, you understand that it's very important that if I have rain and the soil is wet, with all that biomass, it's not going to dry very fast. So to identify the right time to do this work is critical, and we want to incorporate and apply the ASD treatment possibly in the same day to take advantage of all that fresh biomass and all the sugars that are provided by the carbon source. So after plowing, we do disc plow, and often we need also another pass with rototilling to kind of refine the soil, and then we can create false bed. And in this case, we use this cover crop as another test to do another test, and we compared sorghum Sudan grass combined only with water, and then we compared it with molasses and wheat middlings, and we used 2 rates of molasses and 2 rates of wheat middlings. And then, after incorporating everything, we lay out the plastic and again the sensors to monitor everything. And what you see here is the same graph showing how the treatment affected the level of anaerobicity. One thing you can see here, the treatment that reached the lowest level of anaerobicity is really the one with the wheat middling sorghum combined with the full rate of wheat middlings. And molasses here, we see still the fact that molasses is like determining like longer period of anaerobicity. Now here, what you can see in this slide is the cumulative redox potential, and what was achieved with sorghum with plus just water was not enough to say the treatment was effective. But then adding half rate of molasses or half rate of wheat middlings, we could say, especially for this one, we had very good anaerobic conditions. And then, of course, increasing the rate of wheat middlings or molasses, we had even better anaerobic conditions. Now, what I can say here is that these results are determined not so much by the amount of biomass, the amount of carbon that we applied, but really were determined by the carbon-nitrogen ratio of sorghum, molasses, and wheat middlings, and the combination of these three, that we used. With this, like, I would like to say that, again, the carbon source quality, and in particular, the lability of the carbon and the carbon-nitrogen ratio are important factors to determine the efficacy of cover crops as a carbon source. And it's the same thing for other type of organic amendment that we discussed last month in the last webinar. Now, with this, I wanted to I wanted my colleague Kathleen Arrington to start presenting the case study that we have, and because it's really related to the carbon-nitrogen ratio. So I'm gonna give her full control of the presentation now.
All right, well, I'll just start right in. So I'll be talking about one of the experiments at our Penn State Research Station where we use cover crops as a carbon source for ASD. For this experiment, we had 2 research questions. We were interested in whether or not the cover crops alone could provide a sufficient carbon source for ASD, or would they need to be supplemented with an organic amendment? And when we did try supplementing with an organic amendment, we were interested in both the cover crops alone and those with the added amendment, how those combinations affected both the nitrogen supply and the yield of the following vegetable crop. So here's an overview of our timeline for the experiment. The experiment was done 2 years in a row, starting in September, where we planted overwintering cover crops. So here they're being planted in mid-September. Here is what they look like later in the fall. And then at the time, right before terminating them in May, and they were terminated, as Francesco mentioned, with the flail mower and then incorporated into the soil in preparation for the ASD treatment. So the ASD treatment begun immediately after the cover crops were terminated and incorporated, and we have some similar photos of the ASD setup. The ASD treatment for this experiment ran 3 to 4 weeks. So the tomatoes were our vegetable that followed the cover crops, and they were planted in mid-June. And then we assessed the plants in July and August, and then the tomatoes were harvested in September. So each experiment was a full year, and that started in 2021, the fall, and then the first harvest was September 2022. In that same month, we were starting the second year experiment in a field right next to the first. So the cover crops that we used for this experiment, we intended to have 2 contrasting species in terms of plant family and also the carbon to nitrogen ratio that Francesco spoke about. So we had crimson clover as our legume and triticale as a grass, and then we had a mixture of the 2 species. They were planted in mid-September, and this is what they looked like about a month later. So at the time that we were ready to harvest the cover crops or mow them, we sampled them to determine the biomass. And so this is showing for the 2 years, the mixture, the clover, and the triticale, how much biomass was generated. You can see that the mixture and the triticale tended to generate more biomass than the crimson clover. And also that the second year, there was a higher biomass than the first. Even though the planting dates and the termination times were almost identical for the 2 years, the second year winter was milder, and so the cover crops became more mature and resulted in a higher biomass. We took samples from the cover crops and analyzed them for the nitrogen content. And so the crimson clover, typical of a legume, had a higher nitrogen content than the triticale. And in both years, the clover had about twice as much nitrogen as the triticale in terms of percentage. So when you consider those differences in the nitrogen content for both the mixture and the crimson clover, in both years, we're looking at about 100 pounds per acre of nitrogen in the above ground biomass, compared to about 70 pounds per acre of nitrogen in the triticale. As Francesco mentioned in the pot study, assessing the root biomass, but in this experiment, we only measured the above ground biomass. So let's look at what's going to happen to those cover crop residues as they decompose. So for example, these crimson clover residues with the high percent nitrogen, as they start to decompose by the soil microbes, they tend to have enough nitrogen in the cover crop residues themselves to be able that they end up releasing nitrogen into the soil in the form of ammonium and nitrate, which can be taken up by plants. The nitrate is also vulnerable to potential losses to the atmosphere or below the root zone through leaching. In contrast, when you have a cover crop like the triticale that has a lower percent nitrogen, as the soil microbes start to decompose those residues, they often don't have enough nitrogen to meet their own needs, so they need to utilize nitrogen from the soil. And taking up the ammonium and nitrate from the soil can tie it up in the soil microbes, making it unavailable to the following vegetable crop in the short term. So the carbon to nitrogen ratio, we keep coming back to this theme as it being really important in determining whether or not those cover crop residues are supplying nitrogen versus tying it up for the following vegetable crop. So this is the typical range that you would expect of carbon to nitrogen ratios of cover crops, going from 5 to 1 up to 40 to 1. Again, that low C to N ratio corresponds to a high nitrogen concentration. The high C to N ratio means a low nitrogen concentration. And that low C to N ratio tends to have microbes release excess nitrogen to the soil. And the high C to N ratio is associated with microbes tying up nitrogen in the soil. And then our neutral point between on the spectrum is about 20:1 for the C to N ratio. So our clovers tend to fall on the low end, usually in a fairly narrow range from about 10 to 15:1. Whereas triticale has a much wider range of possible C to N ratios. So we measured it in the fall, and it was about 15:1, similar to the clover. By the spring in 2022, it was up to about 28. The clover triticale mix usually has an intermediate value in terms of C to N ratio. So the first year of the experiment, it fell right at that neutral level at about 20:1. Again, I mentioned previously that the second year, the cover crops became more mature. So we ended up with some higher C to N ratios in the triticale and the mixture, potentially leading to some nitrogen tie-up when those residues were decomposing. So I've talked about our different types of cover crop plots that we had. We also had some fallow plots integrated as controls. And then, as I mentioned, we tested one organic amendment, the wheat middling. So Francesco compared some other ones, but for this study, we only used a single amendment. And the wheat middlings have a carbon to nitrogen ratio of 18. So all of these different cover crops applied alone or with the wheat mids had water applied during the ASD treatment. And then we had one other set of controls where everything was the same except that the plots did not receive water during the ASD treatment. So this is another one of those graphs where we're looking at the anaerobicity of the different types of plots in the experiment. This one's from 2022. Again, towards the top of the graph, we're looking at soils that are more anaerobic, and the further you move down, the soils are more and more anaerobic. So our top gray line here is our untreated control, which didn't receive the water, and as expected, it remained aerobic throughout the entire experiment. But now we'll look at a little bit of a comparison among the different types of plots. So within the first week, you can see that the plots that were the most anaerobic were the ones that had the wheat mids applied. So the fallow plots with wheat mids alone, and then also the crimson clover, the mix, and the triticale with the wheat mids added became the most anaerobic at first. As we moved into like 2 to 4 weeks out from when the ASD treatment started, the ones that remained the most anaerobic were the crimson clover alone, even without the wheat mids added, and then the other two cover crops with the wheat mids added. For this year, the tomatoes were planted at 4 weeks, and all of the different types of cover crops with or without wheat mids were anaerobic for at least 3 weeks. Now we'll move on to looking at what is happening to the soil nitrogen levels during and following the ASD treatment. So this is 2022 again. And first we'll look at the plots that only had the cover crops. We're looking at the levels of soil nitrate over time. And just for the different cover crops grown alone, we can see that as right at the time that the ASD treatment is ending and we're about ready to plant tomatoes, the crimson clover clearly had higher soil nitrogen levels compared to the other plots. And so you can see that the crimson clover line is above our fallow control, whereas our triticale is down below the fallow control, indicating that nitrogen is being tied up in those residues by the soil microbes. Now we'll move on to looking at the cover crops with the wheat mids added. And so you can see a similar pattern here with the exception that for the mixture and the triticale, adding the wheat mids that tended to increase the soil nitrogen levels compared to when they were just grown without the wheat mids. We'll look at the same types of graphs for 2023, and we see some similar patterns with the crimson clover having the highest nitrogen supply and also the mixture coming in second there. And when we add the wheat midds, in the second year of the experiment, we had kind of a more dramatic effect where adding the wheat midds is really increasing the soil nitrogen levels for all of the different cover crops and the fallow. So here's a preview of what the tomato plants looked like in August 2022. And a little earlier in the growing season, it's the second year for July 2023. And you can already see some visual differences in how the plants looked when grown after these different cover crops. So we'll cover here the tomato plant biomass, which is just assessing kind of the size of the tomato plants. And this was done about a month after planting them. So you can see in the picture and also in the graph on the left here that the tomatoes grown after triticale tended to be smaller, sort of at a similar level to our fallow control plots here. And whereas the mixture and the crimson clover alone tended to have larger plant size after them. And also specifically for the triticale and the mixture, adding the wheat midds tended to increase the tomato plant biomass. And then we'll move on to the yield of the tomato plants in both years. And here we're looking at the cumulative marketable yield. So first you'll notice that the 2022 yields are quite a bit lower. And at the bottom, we have the different types of plots here. And there's not as much of a contrast with the different cover crops with or without the wheat midds in 2022. 2022 was a different growing season. It was a hotter, drier season, and we had some issues with blossom end rot, so some of these yields are showing not reflecting the total yield. So some of the tomatoes were not considered marketable because of the blossom end rot. So even though that was an issue in 2022, you can still see somewhat of an effect, like a contrast between the tomato yields plants grown after triticale versus crimson clover. In 2023, overall, the yields were higher, and also you can see more of a difference, more of an effect of the wheat midds. So at the dark orange bars here, you're seeing that there's really an effect of adding the wheat midds, for example, to the triticale, increased the total marketable yield, also for the mixture. And even for the crimson clover, there was a smaller increase in the marketable yield when the wheat midds were added. So to summarize our open field experiment, we found that the cover crops varied in their effectiveness as a carbon source for ASD, and also in their ability to supply nitrogen to the following vegetable crop. And we found that the carbon to nitrogen ratio of the cover crops is a key property that influences not only the ASD effectiveness, but also the nitrogen supply in the following growing season. And we found that by supplementing those higher C to N ratio cover crops with the wheat midds, it led to increases in the soil anaerobicity during the ASD treatment, the nitrogen supply at the planting time, and also the plant size and tomato yield. So I only showed 2 different cover crops in this experiment. So now I'm just going to talk a little bit more generally about other species of cover crops. This table was developed for another project, and we're looking at a variety of overwintering cover crops. And these were collected from samples all across Pennsylvania over a 10-year period. So you can see at the top here in the legume group, similar to crimson clover, the other legumes, they all tend to have lower C to N ratios within a relatively narrow range. So you can expect that they would tend to result in a nitrogen supply during and following the ASD treatment. The C to N ratio of the non-legumes are a lot more variable. Sometimes they can supply nitrogen, other times they can lead to nitrogen tie-up. And then in the mixtures, again, there is a wide range sort of depending on what's in the mixture. So if the mixtures contain any amount of legumes, it tends to keep the C to N ratio lower, but the ratio is affected by the percentage of legumes in the mixture. and also by the maturity of the non-legumes. So that I'll talk a little bit more about that in this next slide. This is an example with cereal rye. We found for this previous table that by breaking the cereal rye up into different maturity categories that we could have, it was a better indicator of the C to N ratio. So the picture on your left here is showing an early maturity cereal rye with a C to N ratio of about 20. And then as the rye matures, the C to N ratio gets higher, all the way up to about 40 to 1, where we would expect that nitrogen tie-up as those residues are decomposing. So if you are considering selecting cover crops as a carbon source for ASD, some things to consider. Again, as Francesco mentioned earlier, that it's really important to consider your overall management goals and your planting window that is available for planting the cover crops. But to aim for cover crops having a low carbon to nitrogen ratio right before the ASD treatment to increase that effectiveness and contribution to soil fertility. So to achieve that, you can look to planting legumes or mixtures that are heavy on the legumes. And if you are planting non-legumes, consider terminating them at an earlier maturity to make the C to N ratio lower and better for the ASD treatment and the soil fertility. We found that applying manure to growing cover crops tends to reduce the C to N ratio just because the plants have access to that sufficient nitrogen source. And then also that supplementing cover crops with a higher C to N ratio, supplementing them with an organic amendment that is both low in the C to N ratio and also readily decomposable for the soil microbes is really important. With that, I wanted to say thank you to many people that helped with this project, from our farm crew to a number of grad students, undergrads, postdocs, a wide variety of researchers that contributed to this work, different lab groups at Penn State and also in Florida, and also acknowledging our funding sources for this project. And thanks to Alice and eOrganic for hosting this webinar series. I think that's all we have, and we're happy to take questions or comments if you have any.
Okay, great. Thank you very much. We're going to have a Q&A session now, and I just want to remind everyone that this webinar is being recorded, and you'll be able to find it on the eOrganic YouTube channel within the coming week, along with the past webinars in this series. Okay, yeah, here's a question maybe Francesco can answer. What are wheat middlings?
Yeah, what are wheat middlings? So we use wheat middlings as an alternative, like carbon source that could be combined with cover crops or just used by itself. Wheat middlings is a residue of the wheat milling process. Is like the cheapest, so after you mill wheat and you have flour from that, you have also wheat bran. Wheat bran is like higher value, and then the least valuable byproduct of the milling of the wheat is wheat meats. Usually is used to feed animals. So it still has like a residue of sugar and nitrogen and everything, and is finely ground. So it's very good carbon source for ASD application, and is locally available. So if you want to learn more about wheat middlings, check the webinar we did last time. There's more information in there.
Okay, and do you know the rate that was used for the triticale?
So we definitely have record of the weight, the rate that was used for all the cover crops. I don't remember from the top of my head, and like I don't know if Kathleen has that on hand. I don't, but we can definitely provide that information if the person asking the question would like to know that. We can provide that information.
Okay, do you know what the CNN content of the wheat middlings is?
It can vary, but usually is around 18 to 1. So that's based on the analysis that we have. And I see that there was a question also, like I think is related to the wheat middlings, what rate was used when added to the triticale. I think that we are using as a standard rate the total carbon content. And so like to define the standard rate, we use the total carbon content, and our target is 6,000 kilograms per hectare of total carbon. Now that usually correspond around, like in the case of wheat millings, without, like as it is, without drying it out, is between 15 and 18 megagrams, like metric tons per hectare. But it can vary depending on, what is the total carbon content and the moisture level. Usually the moisture level is pretty low and is a carbon source that could be stored also over time.
Okay. How were nutrients managed? Were there any nutrients applied at the time of ASD application? And do you think that application of mineral source of nitrogen could enhance the CN ratio for species like triticale?
Yeah, so that's a good question. And I would say in this case, I would like to specify that we are working in an organic vegetable production system. And so like we are not using any mineral fertilizer. Of course, like adding a nitrogen source that is mineral fertilizer in a conventional system could change. If you add nitrogen, could change the situation there. So that is feasible in a conventional system. In the specific case of our study, because of our interest in evaluating what is the impact of the cover crops and the amendment that we use on the nutrient dynamics, we do not apply any fertilizer during the growing season. And the only like extra organic amendment that was made was manure, uh, like at the time of the seeding of like right before seeding the cover crop. And I believe the rate there was 50 pounds per acre or something like that. So I see Kathleen is saying yes. So my
Yeah,
memory, I guess, is
I think
good.
50 pounds of nitrogen, I think, per acre applied like right before the cover crops were planted.
Yeah, so one thing that I can say also regarding this is that in all the studies that we have done so far, if you balance well the carbon nitrogen ratio, even in an tunnel system where we have done like double crop, let's say a winter crop like lettuce or cabbage, and then followed by tomatoes, we see the residual fertility of the organic amendment all the way, through the end of the tomato cycle. And we don't have, sometimes we have wonderful crop, beautiful crop without having to add any fertilizer, any supplementary fertilizer.
Okay. In Florida, cover crop establishment in spring can be poor if no irrigation is provided, especially sun hemp. Did you provide irrigation when you were seeding the cover crops?
Okay, yes, that also is a good question. I think that, the answer there depends on, like, so in general, we have not applied irrigation to cover crops, to our cover crops. I would say if you are working in in the condition that described like in Florida, or if you are working in an eye tunnel, irrigation may be necessary to have a good establishment. In our case, we have been working primarily with overwintering cover crops. And so we seed them around, early mid-September, and usually we have rain around that time. And so the establishment is really good. We may have some issue if we delay the seeding, but the seeding date. But drought stress is more a concern for spring and mainly for summer cover crops. And I was showing some pictures of forage radish being affected by drought stress. And that was like spring, summer cover crop in that case. So sometimes you can have this type of issue. And that's why like it is very important, like we cannot provide recommendation of, this cover crop or this cover crop mix will work anywhere. No, you have to really study and understand the dynamic of the rain and the environmental condition in your area and the cycle of the cover crop that you're doing to make sure that you have a good cover crop. And drought stress could be a limitation. Even a week, 10 days can literally like reduce like the biomass that you generate, and that will give space to weeds, and you may have issue in that sense. Kathleen, feel free to add more information if you know.
Oh, yeah, I don't have anything on that, but I was going to go back to the seeding rates. I looked them up while you were answering some other questions. So for the triticale is 120 pounds per acre, which I was just kind of Googling other seeding rates. It does look kind of on the higher side, sort of as you mentioned, Francesco, that you really want to plant on the high end of the seeding rates to really generate a lot of biomass if you're intending to use it for the ASD treatment. And then for the triticale, as part of the mix, it's a lower, we only use 37 pounds per acre. And that was really just to kind of balance the competitiveness of the two species so that we sort of aimed for like an even expression of them in the mix.
Okay. Is there any negative impact on the soil in terms of pathogen buildup if the cover crops used as carbon sources in ASD do not look healthy or are infected by some pathogen?
Yeah, so that is a good question there. I will say we can make some distinction. In general, the pathogen, so in general, cover crops may not be affected by the same pathogen of the cash crop. And we use cover crops to rotate, with cash crop, with the main crop. And in general, we should always choose like species of cover crop that are not affected by the same pest and pathogen issues. The example I made before was you could have a cover crop that, let's say, is susceptible to nematodes, and that cover crop, if you're trying to, if nematode is an issue, you should avoid using cover crops that, can increase the inoculum of nematodes. But in general, if we're talking about, let's say, foliar pathogens that may affect a cover crop, those are not a concern. Like, we are concerned more about soil-borne pathogens in this case with the ASD treatment. I will say in general, like, foliar pathogen may not be a concern when we are using cover crop, simply because they may not be the same pathogen that affect our crop. And Kathleen, you can add to that if you have more comments.
I just want to thank everyone for all the questions. We've come to the end here and remind everyone that you'll be able to find the recording of this webinar and the others on the eOrganic YouTube channel and on the project website. So thank you very much, Kathleen and Francesco, and we look forward to hearing more updates on this project in the fall.
Thank you, Elise and Kathleen and everyone attending, and we look forward to provide more information on this ASD system. Thank you.